We're going to be discussing at this session very important topics that pertain to the diagnosis and treatment of infectious keratitis, and particularly the most difficult forms of infectious keratitis, which are amoeba and fungal infections and viruses. So from this session, what we're going to learn is what are the new molecular diagnostic methods for identifying the disease and how these molecular methods can be applied to better treat a patient based on the type of organism that's infecting their cornea. So the audience will be able to come out of the session with fairly new information. The most promising areas of research in the area of infectious keratitis is going to be looking at the DNA of the organisms that are causing infections and learning why the human body cannot deal with these infectious organisms based on perhaps parameters of their genetic makeup. For example, we have identified that certain organisms that have a certain DNA produce toxins and others don't. And being able to mediate the toxin will probably eliminate the destruction of the tissues in humans from these infections. This year's meeting is full of three areas that are very promising to the treatment of vision problems. Number one is going to be the use of gene therapy to treat many of the diseases that now we know why they occurred based on their genetic makeup. Number two is going to be the use of stem cells to regenerate tissues that have been damaged by disease inside the eye. And the third one is how to analyze big data so that we can learn in terms of population management of diseases. Who is more likely to be having a certain disease in the population that they live in. So I think those three are the top subjects that I see in this year's Academy meeting.